The most pervasive carcinogen in our surrounding environment is ultraviolet radiation in sunlight. Each year, 75 to 80% of cancer diagnosis is contributed to the exposure of environmental carcinogens. The main risk factor for several cancers is the environmental genotoxins. Recently, the association between melanoma with UV exposure was revealed by the highest number of somatic mutation per tumor. The impairment of these carcinogens can be found after DNA being mutated. However, sequencing of skin cancer cells identified the solar signature, which is a pattern of mutation that links UV exposure and melanoma. The two major classes of mutagenic DNA lesion induced by UV radiation are cyclobutane, pyrimidine dimers, and 6-4 photoproducts. The cytosines within the dimers deaminate uracil and thymidine. These UV-induced mutations in melanoma found to be not uniform across the genome, therefore genomic and epigenomic features believed to regulate mutagenesis. However, several published studies suggested the factors that influence the susceptibility to acquire UV-induced lesion. This includes chromatin factors in vitro, DNA transcription factors in yeast, genetic features in mammalian cells. The contribution of carcinogen in mutagenesis to cancer has a relationship between carcinogen susceptibility and the mutation heterogeneity. But factors that regulate localized carcinogen susceptibility have a great effect in carcinogenesis by crucial possibilities for mutation, leading to cause differences throughout the genome that can impact genome stability. In this paper, we present the first genome-wide map of carcinogen-induced DNA lesions in the human genome and find variation in lesion distribution by studying the carcinogen susceptibility of lamina association and chromosomal positioning within the nucleus. For experimental protocol, primary fibroblast cells were irritated with UVC, and DNA was purified using RNase A and proteinase K treatment, followed by immunoprecipitation performance, where single-stranded DNA was incubated with anti-CBD antibody and repaired via photolysis. Then slot plot analysis was conducted. Fluorescent intensity values were analyzed. Then genome-wide analysis was performed on DNA containing UV-induced CBDs. The effects of carcinogen exposure on DNA resulted in broad domains of UV lesion observed between replicates at multiple band sizes, from 1 kb to over 1 MB. Reads were highly enriched in dipyrimidines, with TBT being the most abundant dimer. In particular, the heterochromatin state enriched in retrotransposon lines element accumulates the most UV lesions within the chromatin state in the genome, while the frequency of UV-induced lesion increases proportionally with dipyrimidine content. A positive correlation was observed between a chromosome radial position and mean UV lesion abundance. This model shows that carcinogen susceptibility is elevated within the nuclear periphery and reduced in the nuclear interior. The figure shows as the mutation frequency of individual genes increases within the melanoma sample, so does abundance of UV lesions. Lamina-associated domain enrichment analysis revealed that 77% of genes with the highest mutation frequency are located within LAD. What does these results mean? These results suggest that the origin of mutational heterogeneity in melanoma genome is significantly contributed by the intrinsic properties of carcinogen susceptibility, which is regulated by epigenome architect. Together, these results highlight the contribution of lamina association and chromosomal position to cancer development. In conclusion, heterogeneous mutational spectrum is largely determined by intrinsic carcinogen susceptibility, which is determined by genome architect. Alternatively, a bodyguard hypothesis has been proposed whereby chromatin absorbs genetic injuries from exogenic resources. This study demonstrates that epigenome architect presents a primary barrier to cancer development by regulating susceptibility to carcinogens. Future research may reveal therapeutic strategies to reduce carcinogen susceptibility, thus preventing tumor genesis.